Let's take a look at the digestive system, and in this chapter you will learn the functions and processes of digestion. We'll review the anatomy first, and then we're going to take a look at motility, secretion, digestion and absorption, regulation of gastrointestinal function, and the different phases here, the cephalic, gastric, intestinal phases, and then some of the immune functions of the GI tract. Mass balance in the digestive system means basically input has to equal output. So here are all the input areas into the gastrointestinal tract, and then here are the output areas, and in the end of the day they have to balance each other out. You don't need to know any of the specific numbers here, but all of these things are items that are going into the gastrointestinal tract, and then you need to just the fluid removed from the digestive system, and at the end of the day they have to be equal. So it has to be mass balance in the digestive system. So here are the 4 processes in the digestive system. We have digestion, absorption, motility, and secretion, and we will be taking a look at all 4 of these. So absorption means that you are absorbing out of the gastrointestinal tract whatever you've digested. So the digestion happens through enzymes. So you're breaking down larger food molecules into smaller subunits and smaller organic molecules that can be easily absorbed. Motility means that you're moving the food items forward throughout the GI tract, and secretion can go in various ways. You can secrete into the cardiovascular system, you can secrete back into your endothelium here, or the epithelium, and you can secrete back into the GI tract. Start with an anatomy review of the gastrointestinal tract. We're starting here in the oral cavity. So basically the GI tract is one long tube from mouth to anus, and it's skinny in some places, like here the esophagus, and then it gets larger in the stomach area, and then squiggles around in the small intestine and large intestine, and then here we have rectum and anus, and that's the end of the GI tract. So start to finish would be mouth to anus, one long tube. It's about 10 meters if you were to stretch it out, so that's about 30 feet total length. That's pretty long, one tube. And then we have accessory organs like the liver and the pancreas and some others. So here some of the glands are attached to the gastrointestinal tract. They're not part of the GI tract, but they're accessory glands. So here we're starting with the salivary glands. We have 3 of them, pairs: parotid glands, sublingual and submandibular glands, and they're all secreting saliva. So basically the digestion starts in the oral cavity with the salivary glands providing saliva and some enzymes. Then we're moving esophagus, stomach, which has these 3 parts, which you might remember from anatomy. And then we have a pyloric sphincter. That's a valve that controls how much of the stuff from the stomach goes into the small intestine. Then once we're in the small intestine, we have these 3 parts to the small intestine, and accessory organs, pancreas and liver, main ones. The large intestine is mostly water reabsorption and compacting all the stuff that you don't want to keep. So that would be colon and rectum, and then we have the anus with the external anal sphincter that gives you some control over everything, again because the external anal sphincter is skeletal muscle. Here's an anatomy summary picture of the stomach. We have the 3 regions here: the fundus region, the body, and the antrum, and then here's the pylorus, this pyloric sphincter, the gatekeeper that controls how much of the stomach items is going into the duodenum. Note the foldings right here, these squiggle lines, those are rugae. These are surface foldings that increase the surface area. And here is another sphincter that controls any backflow into the esophagus, which we don't want, because the stuff in the stomach is very acidic, and if you get backflow, it's going to hurt the epithelium here in the esophagus, and people experience that as heartburn. Here's more anatomy review. You should remember that there are always 3 layers to the gastrointestinal tract. You have an outer serosa, that's connective tissue, pretty tough tissue. Then we have a muscular layer, actually 2 muscle layers. One is circular and one is longitudinal muscle, so circular and longitudinal muscle. And then we have the mucosa, which you can subdivide if you want into submucosa and just mucosa. So here will be a submucosa that has a bunch of blood vessels in it. And then here the mucosal layer, which is typically folded to increase surface area. So you have a lot of epithelium right here and different types of glands. Here we are in the stomach. And so you have these rugae, these foldings that are forming in very large gastric pits that are reaching far deep in here. And the lining here, these cells, they're secreting various substances, as we will see. Here's a sectional view of the small intestine, same 3 layers. So you have the serosa, that is your connective tissue, the outer layer. Then we have a muscle layer that is split into circular muscle and longitudinal muscle. And then we have the mucosa that you can split into, well, just mucosa with the surface area extensions right here, these villi that are vastly increasing surface area, which is great for absorption. And then we have the submucosa, which has all sorts of blood vessels. Let's take a closer look at the mucosa. It's created from epithelial cells, lamina propria and muscularis mucosae. And we have modifications throughout the GI tract, which include rugae, such as in the stomach, or villi in the small intestine. So there are these gastric glands and crypts and submucosal glands. All of this is for secretion of different materials. And we'll take a closer look as we get through the stomach and the small intestine. And there's also Peyer's patches of the gut-associated lymphoid tissues, or GOLT, that is associated with the immune functions. Now, the submucosa, that's a type of connective tissue, contains the submucosal plexus of the enteric nervous system and has also lots of blood vessels. Then we have a muscle layer, muscularis externa, that has 2 layers of smooth muscle, and that will be longitudinal and circular smooth muscle. It also contains the myenteric plexus of the enteric nervous system, and then the outer covering of the gastrointestinal tract is serosa. And so this is pretty tough connective tissue that provides the cover for the gastrointestinal tract. Another diagram showing the structure of the small intestine. We're starting from the outside, the serosa, this tough connective tissue layer. Then we have 2 muscle layers, circular and longitudinal muscle. And then we have the 2 mucosal layers, just the inner mucosa, which has usually large foldings for increased surface area, and the submucosa with a lot of blood vessels. So it'll be right here. And very briefly here on the migrating motor complex, it's a series of contractions that begin in the empty stomach and end in the large intestine. So here you can see the arrows as we're going through the gastrointestinal tract right here. Now, more importantly, the term peristalsis. So these are wave-like contractions that move the food forward. So here we're starting at time 0, and we want to move this bolus of stuff forward in the gastrointestinal tract. So you basically contract right here, and that pushes this bolus in this direction. And then as the bolus has moved forward, now it's here, and now we're contracting this area and moving it even farther. So that would be an example of peristaltic contraction. It moves this bolus of food forward. By the way, bolus is a fancy term, I guess, for a chunk of food, and that's what it's called once your food has entered the stomach and is being processed further. You can also segment, and for that you would basically contract here and here, pushing the food in this direction here together, and starts bulging out right here. Then you contract right here and right here, so you're basically smashing what was right there, and now you're moving what was in between the arrows in here in this direction. So then you're bulging out over here, and you can go back and forth, contract, move some stuff here, and then contract here and there, and then you're moving the food sort of going back and forth. And that is really good for mixing and just segmenting of food items. Throughout the gastrointestinal tract, you have this motility, and that's provided by the smooth muscle contractions. So you can have tonic contractions. They're kind of sustained contractions on the order of minutes or hours, and they keep the bolus from moving backwards. So you're moving forward in the gastrointestinal tract. Then we have phasic contractions. They're much shorter, only a few seconds, and that is used for peristalsis, to move the bolus of food forward. It also helps with segmentation mixes. So what I showed you in the previous 2 slides, that was these due to phasic contractions right here, this one and this one. These are all phasic contractions. And then we have slow wave potentials. They originate in the interstitial cells of Kajal. So here will be slow wave contractions of gastrointestinal smooth muscle. We don't need any detail on this, so just kind of take a look at this. As usual, you need to reach threshold before an action potential gets fired. And so if something is below threshold, of course, nothing is going to happen. And here the typical kind of staircase effect if you have multiple stimulation. But more interestingly, to me anyway, is how stuff gets actually absorbed. So the physiology of all of this. And we're going to take a look at what types of cells we have that can really enhance the function here of our gastrointestinal tract. First, take a look at parietal cells in the stomach. So you have here one of these gastric pits, and you can see in this epithelium there's a variety of different types of cells. We're going to focus here on parietal cells. And in a parietal cell here in sort of this tan color, what you're going to do is a familiar equation. We're going to combine water with CO2 to form carbonic acid, which then dissociates into bicarbonate ions and hydrogen ions. The hydrogen ions will be pumped into the lumen of the stomach to contribute to the stomach acidity. And the bicarbonate ions, they leave in a mechanism very similar to the chloride shift that you saw in the lungs, and they go into the interstitial fluids and then in the capillary, and then the pancreas will pick those up and return them to the duodenum in order to neutralize the acid that comes from the stomach. So it's a beautiful system here where you are just recycling everything. So water plus CO2 forms carbonic acid. We're going to separate this carbonic acid into hydrogen ions, which will be pumped into the stomach, and the bicarbonate ions will be going into the blood and then returned to the duodenum via the pancreas, the pancreatic duct, to neutralize the acid that is coming from the stomach. So you have the same, very similar system in the lungs where we had the chloride shift and reverse chloride shift, and you can see even here, same thing. You have also the exchange of chloride for bicarbonate, and here again on the other side as well. So same system basically. Here's a picture of the pancreas, and so the pancreas will pick up the bicarbonate from that reaction, and it will return it back into the duodenum via the pancreatic duct. So it's going to go through here along with a bunch of other digestive enzymes and some other stuff. So anyway, here are some of the pancreatic cells, and you have pancreatic islet cells. They will secrete hormones such as insulin and glucagon. And then you also have this exocrine function of the pancreas, where the materials are secreted into a duct. And then here's the lumen of the duct. And now we're going through the pancreatic duct into the duodenum in this case. And so here will be a pancreatic duct cell. And you will notice that we're going to go exactly the opposite direction of what we just had in the parietal cells in the stomach. So now the goal is to secrete bicarbonate ions into the lumen of the pancreas or the intestine. And so the pancreatic duct, that is, in the lumen of that duct. And because we want to neutralize the acid that's coming from the stomach. And so here we have this reverse chloride shift going on. There is a CFTR channel. CFTR stands for cystic fibrosis transmembrane conductance regulator. And that thing is just like in the lungs, where it's basically a chloride channel. But here we're using it in exchange for the bicarbonate leaving the cell. We're going to invite the chloride in, and that's of course a chloride shift. And you're always dealing with the same equation, always the same thing. So anyway, this principle here is used in a complete recycling system, where we first generate a carbonic acid, separate it into hydrogen ions that are being pumped into the stomach, and the bicarbonate ions are being returned via blood and the pancreas and pancreatic duct back into the duodenum to then neutralize the acid from the stomach. And here is the secretion, chloride secretion by intestinal and colonic crypt cells and salivary glands. So same principle. The chloride here enters into the lumen of the gastrointestinal tract through a CFTR channel, just as before.